Chapter 23, Rebuilding Mitochondrial Efficiency. Mitochondrial efficiency is defined by how effectively the inner membrane can convert substrate oxidation into a contained proton gradient and then into ATP. The machinery of the electron transport chain remains functional across a wide range of conditions, but its output depends on the integrity of the membrane that surrounds it. When the membrane allows uncontrolled proton movement or fails to maintain the organization of its protein complexes, energy is lost during conversion. Rebuilding efficiency therefore centers on restoring the structural conditions that allow the gradient to be generated, contained, and used with minimal loss. The inner membrane must reestablish low permeability. Proton containment is the core requirement for efficient ADP production. As stable lipids replace more permissive structures, the bilayer becomes less susceptible to uncontrolled proton flow. This reduces leakage and increases the proportion of the gradient that is available for ADP synthesis. The change does not alter the capacity of the electron transport chain itself. It improves how effectively that capacity is translated into usable energy. Cardiolipin plays a central role in organizing the electron transport chain. This phospholipid anchors complexes within the membrane and maintains their spatial relationship, allowing electrons to move through the chain in a controlled sequence. When cardiolipin composition is altered or damaged, these relationships become unstable and electron flow becomes less efficient. Rebuilding membrane integrity supports the proper structure of cardiolipin, restoring the alignment of complexes and reducing the likelihood of electron escape. Electron leakage decreases as structural organization improves. When electrons move through the chain efficiently, fewer deviate from their intended path to form reactive species. This reduces oxidative stress within the mitochondrion and limits damage to lipids, proteins, and mitochondrial DNA. Lower oxidative load further supports membrane stability, reinforcing the conditions required for efficient energy production. ADP synthase operates more effectively within a stable gradient. This enzyme depends on a consistent flow of protons through its channel to drive the conversion of ADP to ATP. When the gradient is weakened by leakage, the rate of ADP production declines, and the system compensates by increasing substrate oxidation. As membrane integrity improves and the gradient strengthens, ADP synthase can operate at a higher efficiency, reducing the need for compensatory increases in input. Mitochondrial dynamics contribute to the rebuilding process. Fusion allows mitochondria with improved membrane properties to share components with others, spreading functional improvements across the network. Fission removes segments that remain damaged or inefficient, allowing them to be targeted for degradation. These processes depend on both membrane integrity and energy availability, and they become more effective as conditions stabilize. Energy availability improves as efficiency increases. With less energy lost through proton leakage and reactive species formation, a greater proportion of substrate oxidation is converted into ATP. This stabilizes cellular energy supply, supporting processes that depend on consistent ADP levels, including transport, signaling, and repair. Substrate utilization becomes more balanced under these conditions. When mitochondria operate efficiently, the system does not require excessive input to maintain output. This reduces strain on metabolic pathways and limits the accumulation of intermediate products that can disrupt cellular function. The relationship between substrate availability and energy production becomes more proportional. Repair mechanisms within the mitochondrion operate more effectively as oxidative damage decreases. Enzymes responsible for maintaining mitochondrial DNA and protein integrity require energy and a stable environment. As efficiency improves, these systems can keep pace with damage, preserving function over time. Rebuilding mitochondrial efficiency, therefore, involves restoring the structural integrity of the inner membrane so that electron transport, proton gradient formation, and ATP synthesis operate within controlled conditions. The machinery remains present throughout, but its output becomes aligned with input only when the membrane provides the containment and organization required for efficient energy conversion. Cardiolipin restoration. Cardiolipin defines the inner mitochondrial membrane as a functional surface rather than a simple barrier. It anchors electron transport complexes, stabilizes their orientation, and maintains the curvature required for dense packing of Christie. When cardiolipin is intact and correctly composed, the membrane holds these complexes in close proximity, allowing electrons to move through the chain with minimal deviation. When it is depleted, oxidized, or remodeled into less stable forms, this organization breaks down. Restoration therefore focuses on rebuilding both the quantity and structural integrity of cardiolipin so that the membrane can support controlled energy conversion. Synthesis begins with the availability of appropriate lipid substrates and sufficient energy. Cardiolipin is assembled within the inner membrane using precursor phospholipids that reflect the circulating lipid pool. When stable fatty acids dominate, newly synthesized cardiolipin forms with acyl chains that support tight packing and resistance to deformation. This improves its ability to anchor protein complexes and maintain the local membrane environment required for efficient electron transfer. Remodeling refines cardiolipin composition after initial synthesis. Enzymatic processes replace fatty acyl chains to produce a more uniform structure that matches the functional requirements of the mitochondrion. When the substrate pool contains unstable or highly oxidizable lipids, remodeling incorporates these components, reducing cardiolipin stability. Shifting the available substrates allows remodeling to produce a more consistent and durable form, improving its ability to maintain membrane organization. Oxidative damage represents a primary source of cardiolipin dysfunction. Located adjacent to the electron transport chain, cardiolipin is exposed to reactive species generated during electron transfer. When these species interact with its acyl chains, they alter its structure and reduce its ability to bind and stabilize protein complexes. Damaged cardiolipin disrupts electron flow, increasing leakage and further oxidative stress. Reducing oxidative load and limiting the presence of highly susceptible lipids lowers the rate of this damage, allowing restoration processes to outpace degradation. Crystal structure depends on cardiolipin integrity. The folds of the inner membrane increase surface area and create compartments that concentrate electron transport components. Cardiolipin contributes to the curvature and stability of these structures. When its composition is compromised, cristae become less defined, reducing the efficiency of energy production. Restoration of cardiolipin supports the reformation of these structures, improving the spatial organization of the electron transport chain. Protein interactions are stabilized as cardiolipin integrity improves. Many components of the electron transport chain require direct interaction with cardiolipin to maintain their conformation and activity. When this interaction is disrupted, complexes become less efficient and more prone to releasing electrons prematurely. Re-establishing cardiolipin structure restores these interactions, reducing electron leakage and improving the consistency of energy conversion. Mitochondrial dynamics assist in distributing restored cardiolipin. Fusion events allow mitochondria with improved membrane composition to share lipids and proteins, spreading stability across the network. Fission isolates regions that remain damaged, enabling their removal through quality control processes. These dynamics accelerate the normalization of cardiolipin across the mitochondrial population. Energy production improves as cardiolipin is restored. With better organization of electron transport complexes and reduced proton leakage, a greater proportion of substrate oxidation is converted into ATP. This stabilizes cellular energy supply and reduces the need for compensatory increases in metabolic throughput. Repair processes within the mitochondrion become more effective as oxidative damage decreases and cardiolipin composition stabilizes. Enzymes responsible for maintaining mitochondrial components operate within a more controlled environment, allowing them to preserve function over time. Cardiolipin restoration therefore reestablishes the structural foundation of the inner mitochondrial membrane. By rebuilding its composition and protecting it from ongoing damage, the membrane regains its ability to organize electron transport, contain the proton gradient, and support efficient ATP production. The improvement in mitochondrial function follows directly from a restoration of this lipid framework rather than from changes in the protein machinery itself. Reducing electron leak and ROS production. Electron leak occurs when electrons deviate from their intended path through the electron transport chain and react prematurely with oxygen. This is not a failure of the chain itself, but a consequence of structural conditions that disrupt controlled transfer. The inner mitochondrial membrane must maintain both the spatial organization of complexes and the confinement of charge to ensure that electrons move stepwise through defined carriers. When this organization weakens, electrons escape, forming reactive species that do not contribute to ATP production and instead propagate damage within the membrane. The stability of electron flow depends on how closely the complexes are arranged. Cardiolipin anchors these complexes and maintains their alignment, allowing electrons to pass efficiently between them. When cardiolipin structure is compromised, the distance and orientation between complexes change, increasing the probability that electrons will not reach their intended destination. Restoring membrane composition reestablishes these relationships, reducing the likelihood of premature electron transfer to oxygen. Proton gradient containment influences electron behavior. A stable gradient creates back pressure that regulates the rate of electron transport. When protons leak across the membrane, this pressure decreases and electron flow accelerates in an uncontrolled manner. This increases the chance of electrons escaping the chain. By reducing membrane permeability and restoring gradient containment, electron flow becomes more controlled, lowering the rate of leakage. Lipid composition determines susceptibility to secondary reactions once reactive species are formed. Membranes rich in highly unsaturated fatty acids provide multiple sites for these species to initiate chain reactions. As stable lipids replace these structures, the number of reactive targets decreases, limiting the propagation of damage even when small amounts of reactive species are produced. Antioxidant systems within the mitochondria intercept reactive intermediates before they can interact with surrounding lipids. These systems rely on both availability of antioxidant molecules and proper localization within the membrane. When oxidative load is reduced and membrane organization improves, these defenses can neutralize reactive species more effectively, preventing accumulation. Electron input must remain matched to the capacity of the transport chain. Excess substrate delivery increases the rate of electron donation, which can overwhelm the system when structural conditions are suboptimal. As mitochondrial efficiency improves, electron flow aligns more closely with the capacity for controlled transfer, reducing the pressure that leads to leakage. Mitochondrial dynamics contribute to reducing electron leak by maintaining a population of functional organelles. Fusion allows mitochondria with stable membranes to share components, improving overall performance. Fission isolates regions where electron leak remains high, enabling their removal through quality control mechanisms. This maintains a network that operates with reduced variability in electron transport. Energy production becomes more efficient as electron leak decreases. Fewer electrons are lost to reactive pathways, and a greater proportion contributes to proton gradient formation and ADP synthesis. This reduces the need for compensatory increases in substrate oxidation and lowers the overall oxidative burden on the system. Reducing electron leak and reactive species production therefore depends on restoring the structural integrity of the inner mitochondrial membrane and aligning electron flow with its capacity. By improving complex organization, maintaining gradient containment, and limiting susceptible substrates, the system reduces the frequency and impact of electron deviation. The result is a more efficient conversion of substrate energy into ADP and a lower rate of membrane damage from reactive intermediates. Re-establishing gradient integrity. The proton gradient across the inner mitochondrial membrane represents stored energy in its most usable form. Electron transport builds this gradient by moving protons into the inner membrane space, creating both a concentration difference and an electrical potential. ADP synthesis depends on forcing those protons back through a controlled pathway. When the membrane holds the gradient tightly, the flow is directed through ADP synthase and energy is captured efficiently. When containment weakens, protons return through the bilayer or through nonspecific pathways, and the stored potential dissipates without producing work. Re-establishing gradient integrity means restoring the membrane conditions that prevent this uncontrolled return. Permeability is the primary variable. The inner membrane must resist proton movement except through defined channels. This resistance is determined by lipid packing density and the absence of structural defects that allow charge to bypass ADP synthase. As stable lipids replace more permissive structures, the bilayer becomes less conductive to proton flow, and leakage declines. Even small reductions in permeability have large effects on gradient stability because the system operates continuously. Lower leakage allows the gradient to build and persist with less input from electron transport. Electrical properties of the membrane contribute to gradient maintenance. The separation of charge across the bilayer creates an electrical field that influences how protons move. A stable membrane maintains this field without fluctuations caused by uncontrolled ion movement. When permeability increases, other ions cross more freely, dissipating the electrical component of the gradient. Restoring structural integrity limits this movement, preserving both the chemical and electrical components that together define the gradient. The organization of the electron transport chain affects how efficiently the gradient is generated. Complexes must be positioned so that electron flow is continuous and proton pumping occurs in a coordinated manner. Cardiolipin stabilizes this arrangement, ensuring that protons are moved into the intermembrane space at a rate that matches the capacity of the membrane to contain them. When this organization is restored, gradient formation becomes more consistent, reducing fluctuations that would otherwise weaken overall integrity. ADP synthase depends on a stable gradient to operate efficiently. This enzyme converts the potential energy stored in the gradient into chemical energy in the form of ATP. When the gradient is inconsistent, the flow of protons through ADP synthase becomes irregular, reducing the rate and efficiency of ADP production. As gradient integrity improves, proton flow becomes steady and directed, allowing ADP synthesis to proceed at a rate that reflects the actual capacity of the system. Substrate oxidation becomes more efficient when gradient integrity is maintained. With less energy lost to leakage, the system does not require excessive input to sustain ADP production. This reduces the burden on metabolic pathways and lowers the generation of reactive species associated with high throughput conditions. The relationship between substrate input and energy output becomes more proportional. Mitochondrial dynamics support gradient restoration by distributing stable membrane components across the network. Fusion events allow regions with improved integrity to influence others, while fission isolates areas where leakage persists. This process gradually increases the proportion of mitochondria capable of maintaining strong gradients. Energy-dependent maintenance processes benefit from improved gradient integrity. Ion pumps, transport systems, and repair mechanisms rely on consistent ADP availability. As ATP production stabilizes, these processes operate more effectively, reinforcing the structural conditions that support gradient maintenance. Re-establishing gradient integrity, therefore, involves restoring the physical properties of the inner mitochondrial membrane so that protons are confined and directed through controlled pathways. By reducing permeability, preserving electrical stability, and maintaining the organization of electron transport complexes, the system rebuilds the conditions required for efficient energy conversion. The gradient becomes a reliable store of potential energy rather than a transient state that dissipates before it can be used. Recovering energy consistency. Energy consistency is defined by the ability to produce ADP at a stable rate under varying conditions without requiring continuous compensation. The system does not fail because it cannot generate energy. It fails because production becomes variable, forcing constant adjustment in substrate use, signaling intensity, and cellular behavior. This variability originates at the membrane level, where inefficiency in gradient containment and electron transport introduces fluctuations in output. Recovering consistency requires restoring the structural conditions that allow energy production to proceed at a predictable rate. Mitochondrial membranes must maintain stable gradients over time. When proton leakage is reduced and electron flow is controlled, the gradient does not collapse between cycles of ADP synthesis. This allows ADP production to proceed continuously rather than in bursts followed by depletion. The result is a steady supply of energy that supports processes requiring constant input, such as ion transport and protein turnover. Substrate utilization becomes aligned with demand. In an unstable system, increased leakage forces higher substrate oxidation to maintain output, creating periods of excess input followed by relative shortage. As efficiency improves, the amount of substrate required to sustain ADP production decreases, allowing intake and utilization to remain proportional. This reduces fluctuations in circulating substrates and stabilizes metabolic pathways. Signal-dependent energy demand is met more effectively under consistent conditions. Cellular signaling processes require ATP for activation, propagation, and termination. When energy supply fluctuates, signaling becomes inconsistent, with some pathways receiving sufficient support while others do not. Stabilizing ADP production allows signaling processes to proceed without interruption, improving coordination within and between cells. Ion gradients are maintained with less effort. Pumps that regulate sodium, potassium, calcium, and other ions depend on ADP to sustain differences across membranes. When ADP availability is stable, these gradients remain constant, supporting electrical stability and transport processes. This reduces the need for compensatory activity that would otherwise increase energy demand and introduce variability. Mitochondrial dynamics reinforce consistency. Fusion distributes functional components across the network, allowing mitochondria with stable membranes to support those that are less efficient. Fission isolates regions that continue to produce inconsistent output, enabling their removal. Over time, this process increases the proportion of mitochondria capable of maintaining steady energy production. Repair and maintenance processes operate more effectively when energy supply is stable. These processes require continuous ADP input to replace damaged components and maintain structural integrity. Variability in energy production limits their effectiveness, allowing damage to accumulate. As consistency returns, repair mechanisms keep pace with degradation, preserving function. Hormonal and neural regulation align with energy availability. Many regulatory systems adjust activity based on perceived energy status. When production is inconsistent, these systems receive conflicting signals, leading to over- or undercompensation. Stable ADP production provides a reliable indicator of system state, allowing regulatory pathways to maintain balance without excessive adjustment. Temporal coordination improves as energy becomes consistent. Processes that depend on timing, such as metabolic transitions and cellular cycles, require predictable energy availability. When ADP supply fluctuates, these processes become irregular, leading to inefficiency and interference between pathways. A stable energy baseline allows these processes to proceed in sequence, improving overall system function. Recovering energy consistency, therefore, involves restoring the membrane conditions that allow ADP production to match demand without fluctuation. By reducing leakage, stabilizing electron flow, and aligning substrate use with output, the system reestablishes a steady energy supply. This consistency supports transport, signaling, and repair processes, allowing the body to operate without the need for continuous compensation and adjustment.